Thank you for the invitation. I'm very glad to be here and have the opportunity to present some of the work that we actually support internally in our team at Roche and also together with academic collaborators. So, very briefly, together with enthusiast scientists around the microbiome, we have a focus team that our main objective is basically to accelerate the drug discovery and clinical development, basically by co-creating technologies around the microbiome and expanding the interest across Roche disease areas. And why the microbiome could be very interesting to accelerate the drug discovery pipeline is basically it targets and increase the probability of technical success in some of our clinical trials, in particular by addressing three key points. And the microbiome, we strongly believe that it may help us to have a better patient stratification and try to predict basically the treatment response. Also, it helps us to accelerate development in time by surrogating endpoints, for instance, endoscopy or other very laborious endpoints. And in addition, it also brings a patient-centric benefit, such as the prediction of disease a bit earlier or onset of the disease. So with that said, I would like today to present you in a high-level description some of the recent work that together with the lab of Nicolas Segara and my previous lab, Thilo Strovik, we were able to identify and expand all this diversity associated in the previous known genus Prevotella copri, but now rename it and relabel as a Segatella copri complex. Um, so very briefly to start off something that most of the audience is very familiar, but I just wanted to mention here that these are some of the main questions and most repetitive questions that we receive internally from our clinician colleagues or clinical scientists. And it's basically how many species are present in our gut microbiome. And recently it has been shown that around 270 species can be detected by ultra deep sequencing. And I think that is very much in line with some of the numbers that we observe in some of our studies. In addition, it's also highly asked, like if we know already the full catalog of microbes that we may have, and also whether how is the sharing DNA identity between individuals. And it's actually very different if we compare the identity at the level of nucleotides. So basically, there is an overlap of 10 to 20% between individuals, meaning that the DNA information that is in your gut is very particular and very unique. It may change through lifetime and also is very stable and resilient. In addition, is shaped by what it has been previously identified, like geographical location strongly shape the gut microbiome communities, and factors such as diet, age, sex, or medications may strongly shape and affect the gut composition. In addition, some of the main dominant phyla, people sometimes are interested in how these members are renamed, or basically the abundance of the most abundant taxonomic bacteria. And we can find that Bacteroidetes is Bacillota are among the most abundant members of the gut microbiome. To just a high-level description of what it has been found. Besides this high complex environment, I usually try to tell people that although the gut microbiome is largely diverse and complex, it seems like there is some order in this chaos, and this has been reported like in one of the first large observation in the gut microbiota cohorts, and it's basically the concept of the gut prototype. So basically, it refers that human gut microbiome can be basically grouped in three different clusters based on the most abundant bacterial genera, and those genera are basically Bacteroides, Prevotella, and Ruminococcus. But after almost 10 years of research, we know that this is not as discrete clusters, but basically are more like continuous landscape where an individual's gut microbiome can have a different content of Ruminococcus, Bacteroides, or Prevotella, and the diversity can be moved in this sort of landscape. And the main question is, what are the main environmental factors that may shape this composition? And it's already known that geographical location may have a strong impact, such as diet as well, and industrialization is one of the main drivers of these two predominant groups, Bacteroides or Prevotella. But the main question that remains is like, what are the implications in terms of human health? And I think that at least for the Prevotella lichen serotype, there has been different associations to health and disease, and this is one of the main examples. So in health, it's known that patients with a high Prevotella radio may respond faster and also have a better response upon diet intervention. And also those patients with a high Prevotella radio presented like an improvement in glucose metabolism. On the contrary, in disease, Prevotella has been associated with a high risk of developing rheumatoid arthritis, and also in mouse models, there is a predisposition for an enhancement of colitis. So some of the reasons of this sort of contradictory outcomes, we strongly believe that is the lack of reference isolates, and also it may be explained by the unknown strain level diversity in this genus. So that's why we decided to go deeper in the characterization of Prevotella, or now rename it as a Segatella copri. And because, guys, it's like one of the second and most abundant bacteria in the gut microbiome, and after 10 years was not like really well known, the diversity. And this is what was known so far. When Prevotella expand, here you can spot some of the data sets where Prevotella was highly prevalent and abundant. Here in light orange, when Prevotella is present, tend to really dominate the gut communities. And it was until a few years ago that it's one of the most elegant and beautiful papers in the gut microbiome. I personally like this paper from Nicolas Segara's lab, and basically using 1,000 high quality metagenomic assemblies, they identified that actually Prevotella was conforming 4 different groups, or what they call clades. And in the past, Prevotella was thought to be just one single species, but it turns out that there are like 4 different ones. Furthermore, in the lab at the Helmholtz Institute with Thilo Strohwijk, we were trying to understand what are the determines, and also what are the factors that allow Prevotella to have this overexpansion that you can see in orange. And we were using mouse models and diet intervention experiments to understand what are the key genetic elements that allow Prevotella to expand in the gut. And we found that basically complex polysaccharides from high fiber diets such as arabinoxylan enables the expansion of Prevotella in the gut, and also to dominate and outcompete other species. But more recently, when we wanted to translate and move into the human space, we started like a strong culturomics and metagenomic efforts, and we start observing that there were some isolates that didn't fall in these 4 clades that were previously described. So it was an opportunity to come back to Nicola and start discussing a potential collaboration to expand the screening and the study of the complexity in terms of diversity of these Prevotella or Segatella species. And we use basically targeted isolation. In this case, we also use long sequencing using PacBio, and also we screen all public genomic databases in order to have a more comprehensive overview of the genetic diversity of Prevotella. Furthermore, we perform phylogenetic analysis to understand these clades and distribution at species level, and also we perform meta-analysis to having already that diversity resolution, we wanted to understand if there were some particular associations with disease. And this is basically the first outcome of this study, and was high kudos to Edo, who was working with the phylogenetic reconstruction of the Prevotella copri complex. And he basically took more than 10,000 genomes, and we found that there were more than 4 clades, and basically the diversity can be summarized in 13 distinct species or clades. And when we try to see where those clades were highly associated, we observed that 9 of those clades were strongly associated to the human gut, and 4 of them were also coming from non-human primates. Then using long read sequencing, using PacBio and really deep sequencing of some of the isolates and representative species of each of these clades, we observed something very unusual, and this was the presence of a large extrachromosomal element that it hasn't been described before in Prevotella. And maybe for some that are familiar of these large extrachromosomal elements in gut commensals, we have previously the report of the lag phages. This is one of the most largest extrachromosomal elements that have been described in this group of bacteria, in particular in Prevotella or Segatella. And this is very interesting observation because the size of this element is almost 20% of the size of the genome of Prevotella. In addition, we found the list, and that was a new element that was identifying chromosomal elements present in Prevotella. It can span up to. And some of the interesting features that we found in those large extrachromosomal elements basically were the presence of ribosomal RNA operons, the presence of polysaccharide utilization loci and caseins, and those elements were full of toxin and detoxin systems as well. The real function or implication of these extrachromosomal elements in health or disease is not well known. And also, as I would like to highlight in this picture, here presented in these dot plots, those elements encode several unique and accessory proteins. So they are a bit different from what it can be found in the main chromosome. We wanted to take this further, and basically we wanted to understand how is the distribution of these clades across most of the most common publicly available data sets, and we distribute them in 2 groups, like the westernized gut microbiome data sets versus the non-westernized. We also have the opportunity to study this new resolution of the Segatella copri complex into Asian samples and also non-human primates. And basically what we could observe from here is that westernized samples have a particular abundance and prevalence of the species of clade A, and some of them are losing basically the diversity of other clades. We look this in particularly for these 9 clades that are associated with humans, and interestingly, we also look at the coprolites, and these are really valuable samples that are dated from more than 5,000 years ago, and we found that like really several years ago, humans carry up to 8 different species of Prevotella. And interestingly, some of the coprolites that have been found in South America and North America, they carry 4 of the Prevotella species. moving forward, having this nice resolution at clade level, we wanted to understand whether there was a particular association with the human diseases. So therefore, we took more than 4,000 samples and we assessed and screened the presence of these Prevotella clades in 12 different diseases. But interestingly, we didn't find very conclusive results in terms of the association with disease. We found that some of the Prevotella species are strongly associated with male sex, but they are not associated with particular diseases such as rheumatoid arthritis or IBD. And in a way, it makes sense because this is a commensal that has been associated with the human gut for so many years, and the particular role or hypothesis that maybe some strains are associated with disease, I think that are not very very supported by the current data. So I think that the Prevotella is more like in the side of a normal commensal rather than a pathobiont. And second, we also have the opportunity to look at the more than 1,000 samples that have been nicely followed up in this predict cohort. And there we have like very particular cardiometabolic measurements of these individuals. And we found that the Prevotella is strongly negative associated with visceral fat, meaning that it seems to be associated with low visceral fat in those individuals. And some of the strains of the clade A and the clade C are strongly associated with cardiometabolic biomarkers such as high density lipoprotein, which is basically the good cholesterol. And this is very important because now we are in the age of the development of therapeutics for cardiometabolic disease, and having this such a good resolution in this Prevotella group may help us to understand many different outcomes in those trials. I would like to wrap it up here, and I hope that this high-level description and this story convinced you that having a good characterization of gut commensal is a space for developing novel biomarkers. While we were doing this work, we also found novel interesting biology, such as the LEAs, or the large extrachromosomal elements in Prevotella. And we will keep an eye on this particular association with the improvement of glucose response. Very last comment, the resolution of the Prevotella clades are now part of the Metaflam4, and we are very happy to share these results with the community. Very finally, where do we stand right now? We are exploring and expanding this new resolution of in these commensals and using new tools in order to have a better prediction of patient response, and also trying to understand the progression in complex diseases. And I personally believe that just taxonomical profiles maybe are not good enough to have like clinical level predictions, but we are just testing and evaluating whether using a multi-omics integration with functional profiles and also clinical data may allow us to have a better resolution. This one you already saw. I really encourage you and to move into standards and use the new taxonomy. And finally, I would like to acknowledge all the teams that were involved in this fantastic collaboration, in particular Nicola, who is part of the International Forum, and especially Aitor, who was doing this titanic labor and task, putting all data together and coordinating the teams. So with that, I would like to thank you for your attention and the opportunity to present my work here. Thank you.